Welcome to this introduction to FAIR for Data Management. I'm Cristina Furlan, and I'm a lecturer at the System and Synthetic Biology Department at Wageningen University and Research. You are a PI or a new lab member wanting to continue the project of a postdoc who left the lab. You contact them and you receive a prompt reply to look in folder X on hard disk Y for the data you wanted. But once you open that hard disk, you find this mess. I'm sure the data is there, but where? In this video, we will describe the principles controlling good data management based on FAIR. So finding or using data within your lab will no more be such a struggle. The short anecdote of the messy hard disk exemplifies very clearly why we need to organize well our data. But luckily, we are not starting from scratch. We are already doing it well in the wet labs. We use lab journals or electronic lab journals to describe data, experiments, and none of you will throw their tubes in the freezer just labeling them sample 1, sample 2, sample 3. The same should be done for data. Let's consider you are involved in a project in which you are studying bacteria that are growing in a biofilm. You'd like to dissect the development of this community via multiple omics approaches. You collect genomics information, then transcriptomics at different time points. Then you further analyze some key time point samples via proteomics. And finally, again, all your sample for each time point for the metabolomics. We additionally do this analysis both for a wild type and the mutant strain, as exemplified by the blue and green labels. When you get back the data from the company performing the RNA-seq analysis, for example, you receive files, like the one in the figure. You receive many of them. Think those are only the transcriptomics files of your analysis. So we need to properly store and annotate those RNA-seq files in the same way you would do for storing your tubes in a freezer. So you need to create relationship between the files and the experiments they relate to. In this way, you can keep track of what was done to the sample. So together with the files, we need to store the metadata. This means all the protocols, processing steps, instrument used, statistical analysis done, parameters or thresholds involved, software used, who is responsible for the experiment, etc. We need to do this following a certain structure. This structure is given by the FAIR principle for data management. FAIR is an acronym that stands for findable, accessible, interoperable, and reusable. Data should be findable. They should have unique identifiers and are extensively described with metadata so that they can be searched. They're stored not on an old postdoc hard disk, but can be found on local servers and internet repository. Data should be accessible. Accessibility implies that those entitled to access the data can do it both today or in the future. So there are clear licenses and clear rights to access the data. Interoperable relates to the data being in usable format. It links to the idea that data should be understandable, not only by individuals, but also by computers. In this way, it allows the integration with existing knowledge, metadata. Finally, reusable. Reusable ensures the data remains available for future results. This is possible if the data is reliable, and to be so, the data and associated metadata is complemented with detailed description on how the data has been generated and processed. This refers to what is called the provenance of the data. How can you make sure your data is fair? You can by following the minimal information requirements. These are standards often enforced by submission platforms. For example, if you submit your proteomics data to PRIDE, the Proteomics Identification Database, they have some minimal requirements for the type of data and metadata that needs to be available so that you can deposit your data in the repository. Those, for example, are the raw files and search results file, the experimental design table, etc. So it is important that when you set up a project, you actually try to make your data fair by design so that you can actually put in place all this pipeline and you think about the metadata even before you start actually doing the wet lab experiments, for example. If you're still not convinced about the need of fair data management, you should be aware that nowadays publishers increasingly demand that data becomes available to push reproducibility for scientific discoveries. Plus, governmental agencies and funders increasingly demand data management plans and concordance with fair data principles to be able to support your research. To recap, in this video, we have described why you need to store properly your data. I showed you the importance of having metadata coupled to your data files to describe your results. Then we looked at what principle you should follow for proper data management. Those are collected in the acronym FAIR: findable, accessible, interoperable, and reusable. If you wish to learn more about how to organize your data and make it fair, please refer to our extended seminar in the link in the resources and to our manual. Now, there is much unjustness in the world, but at least for your data, go and make it fair.